Good afternoon all. So today we are going to discuss about use of automated machines in microbiology laboratory. Here, microbiology has always been very traditional and also very labor intensive, with the view that automations was for other disciplines but not suitable for microbiology. It was an old myth that it is not suitable in microbiology. Over the last few years, however, new and improved automated technologies have provided solutions to the challenges which we are facing today's microbiology lab. The first stand-alone automation for the micro lab was introduced in 1950s, with the initial systems primarily designed for blood cultures, TB, antibiotic susceptibility, and biochemical based identification. It wasn't until 2006 that first true bacteriology automation was introduced with barcoding of dishes, inoculation, moving track systems, automated incubations, and digital imaging. And this was a new event which takes place after the 2006. Like many other industry advancements, laboratory automation is designed to increase efficiency, streamline processes, and deliver high quality, consistent results with less time. Today, automation is a complex integration of computers, robotics, liquid handling, processing, and other combined technologies. Automation of routine procedures such as dedicated workstations and software to program instruments has already impacted laboratories worldwide, with repetitive tasks such as pipetting and transporting plates and various types of assay it being the first to be automated. So what's driving the shift to microbiology automation? Each lab probably has its own issues, but the main factors are the continued pressure on reducing cost while increasing the productivity, turnaround time, and result reliability. The current trend is towards merging small labs into large superb labs, which was considered to be the most cost-effective and efficient way to process samples, and these have the advantage of creating centers of excellence in terms of expertise. With the shift from the country-specific accreditation to the more demanding international ISO 15189-2012, automated systems are ideally suited to meet the accreditation requirements by automatically monitoring each step of analysis, and it's retaining the data for later access. Recruiting and retaining qualified, experienced staff, especially with a trend towards 24/7 working. It is also an issue for many labs, so again automation can step in. So what steps can be automated? Which are the steps included in the automation? First one is media preparation. It is perhaps the most well established and long standing area that can be automated. It is media preparation. However, now often labs will not see this as a core activity with all the associated validations and the QC and will buy in ready to use media. Specimen preparation and also the planting, inoculation or streaking plates. They are most fully automated inoculation and streaking systems require liquid transport swabs or liquid samples. Specimens can be loaded into the racks and then loaded onto the instrument. Alternatively, samples can be added to the turntable for continuous loading. And the sample is scanned and the system will know how to process the specimen and what plates are required. After vortexing, means shaking, the required plates arrive ready barcoded so that they can be tracked and traced throughout the process. The plates are then planted or inoculated or streaked depending on what was specified for that particular specimen. A HEPA environment ensures no cross-contamination. A specific streaking patterns can be pre-programmed and achieved by robotic loop. However, robotic technology is most common. This results in a consistent, reproducible inoculation and streaking pattern, and it produces single colonies more often than by a manual process. So manual process is very time consuming nowadays. So most of the labs have converted to automations. System will include a monitoring step to ensure that some sample has indeed been taken up by the pipette or loop. Inoculated plates can then be sorted according to the required atmospheric conditions and the temperature and transported by conveyor belt to the appropriate incubators. Any non-liquid or other specialized samples can be done in a semi-automated fashion whereby the technician prepares the plate, which then goes back into the system with a bulk of samples. Artificial intelligence. The artificial intelligence can be applied to screening and interpretation of plates following incubation. Algorithms can be adjusted to meet a particular lab's requirement to enable the automated screening of non-critical plates depending on visual appearance, sample or patient histories. This results in the vast majority of plates being automatically read and recorded without the need of any technician intervention. Incubation. As each plate is barcoded on the way to the incubator, it's scanned so incubation start time is registered and how long that plate will need to be incubated before going to the plate reader. Plate reading and interpretation. After incubation, plates are automatically moved to the image analyzer for reading and may subsequently be returned to the incubator if necessary. This means plates get exactly the correct incubation time even if due for reading during the night if the lab is 24/7. The barcode on the plate, it contains information on which camera and lighting settings are required to take the images for that particular plate. Even chromogenic plates such as for MRSA can be automatically read and interpreted. The whole plate set from a patient can be put together on one screen for viewing together in one place. So secondary plates such as antibiotic sensitivities can be seen with the primary plates or the image from day 1 can be viewed with the day 2. Images can be saved for the later reference or auditing purposes. Looking at plates on a screen is probably one of the most significant changes that automation brings for the biomedical staff who are used to hold a plate, seeing it in 3D and maybe quickly doing some basic biochemical test. But plates can always be called up to the workbench for the examination by the eye, and as staff gain more confidence in the digitalized system, they will most likely need to only call up those plates that are necessary, leaving the bulk routine plates to be handled by the instrument. Antibiotic sensitivity testing. The inoculation and streaking modules are able to produce seeded plates for sensitivities. However, relevant antibiotic sensitivity discs need to be added using traditional disc dispensers. These plates can be returned to a workbench for the disc to be added. Next is identification. It is typically performed by matrix-assisted laser desorption or ionization time of flight, the short form MALDI-TOF. MALDI-TOF has been one of the most greatest advances in microbiology in recent years due to its speed of result. Together with a low cost per test, it easily outperforms biochemical based approaches. Most MALDI TOF will sit near or immediately next to an automated system, and some systems can use a loop to seed the MALDI TOF target plate automatically. This is the picture of MALDI TOF. This helps to perform antibiotic susceptibility testing, microorganism identification, subtyping, etc. How is the automation different? In most inoculation and streaking systems that are fully automated, the samples first need to be in a liquid format, that is, a liquid transport or swab or urine, although other types of specimen can fit in later in the process. The common perception is that digital imaging can be used to make a determination. In fact, it is used to sort the plates, which may be of interest to do further work or sensitivity testing. The others can be sent to discard without being handled by a biomedical scientist. There will always be some plates that may require a visual check by the laboratorian prior to doing any further work being performed. With centralized processing and reading gets away from the traditional specialized benches or areas, staff can easily access all the data from a particular sample and compare on one screen. It also frees trained, experienced staff from doing dull, repetitive tasks that can be usefully employed in using their skills and knowledge where it is most needed in the unusual results rather than the routine ones. Now look at the advantage of lab automation. It includes increased productivity, more samples processed per patient, a move away from batch processing to continuous even 24/7 processing, the ability to handle surge demands, remote reading and access to image of plates and organisms, assurance that the sample is processed correctly with the right plates and the incubation conditions, ability to view the whole patient plate set and historically plate sets, reduction in the technical and transcription errors, improvement in traceability and fully audit trials including reading process, images available for retrospective and training purpose. So these are all about the automated machines in the microbiology laboratory. Thank you. Also, I will upload the important machines which are used. Thank you.